So, good evening, everyone. A very warm welcome to all of you joining us for today's session on demystifying biosimilars. I would like to begin by introducing our moderator for today, Dr. Shohini Ghosh. Dr. Ghosh is a global executive MBA alumna and topper of the batch of 22, and she's had a long stint in pharma working at the R&D center at the Torrent Pharmaceuticals before joining Ahmedabad University, where she's currently the chief of staff for the vice chancellor. Thank you for moderating the conversation today, ma'am. Over to you.
Thank you, Mehak. And good evening, everyone, and welcome to today's webinar, Demystifying Biosimilars. While there's a rising need for affordable yet advanced healthcare solutions, biosimilars are reshaping access to life-saving medicines worldwide. In this talk, we will explore what biosimilars are, why they matter, why they're so important, and why their development is critical and the science behind it. So please join me in welcoming Malini S. Malini is the Senior Scientific Manager at Biocon and has joined us today to speak on demystifying biosimilars. She is also a dear friend and a fellow GEMBA alumnus from the batch of 23. Thank you, Malini, for giving your valuable time to us for this session. To introduce Malini further, she specializes in bioassay development for monoclonal antibody biosimilars. With deep expertise in biologics and analytical development, she plays a key role as a subject matter expert in advancing high-quality biosimilars. A passionate data science enthusiast, Malini combines her scientific acumen with data-driven insights to drive innovation in the biopharmaceutical space. Thank you, Malini, and over to you.
Thanks, Shohini, for the warm welcome. Good afternoon, everyone. So today we'll be talking about biosimilars. So before we jump into the biosimilars, this is a question that I think most people have who have seen either their friends or families in any condition which requires hospitalization or a disease affecting them. So is the treatment or the therapeutics truly affordable, accessible to everyone? Is a question which we all have to ask ourselves, especially the targeted therapeutics, which are more of specialized ones. They are extremely expensive and make it much more challenging for people in terms of affordability and accessibility. So keeping that in mind is where the answers to some of these targeted therapies come in as biosimilars. So let's talk about biosimilars and demystifying biosimilars for all of us. So if any of you do a quick search or ask ChatGPT or any of the AI engines, what is a biosimilar? You will see that the definition that most commonly would be thrown up is that it's a biologic medicine that is highly similar to a licensed reference product with no meaningful differences in terms of safety, purity, and potency. But when we say a biological medicine, what is a biological medicine? These are nothing but drugs which are made from living organisms. It can be bacterial cells, yeast, or even mammalian cells. So some of the living organisms are coded in to make the drug that is of our interest. That is what is basically a biologic. And when originator makes it, they usually have a patent around that. So after the patent period is expired, there are other companies which can make the same drug and sell it to the patients as well. So this is where the biosimilars come into picture. So we all are aware of what generics is. So if we look at this, this is, say, the original product which was made by an innovator. Generic would be an exact copy of the drug which was made by the innovator. You will not see any differences in terms of its structure or activity, or in any perspective, you would not see a difference between the originator drug and a generic drug. But when we come to biosimilars, These are highly complex molecules which have a very huge molecular weight and structural complexity, functional complexity as well. So if the original product looks like this, the biosimilar would also look like this. So in terms of appearance, on a high level, it does appear similar. But there are minor differences possible because these are produced by living organisms. So although these are similar, they are not exactly identical. And because the process of making biosimilar is much more targeted in terms of we know the outcome, we know the structure, we know what is expected, and it is only about how to make the product exactly the way it is in the originator case. So that's where the affordability comes into picture. But because it's such a huge molecule, it is not possible that it would be identical. And since there is a living system which is making it and not a fixed chemical process like in the case of generics. So why are biosimilars important? If we think about it, it is about bringing better access to life-saving medicines to the patients. It stimulates market competition and innovation in terms of, say, the innovator makes a product in a certain way. The process in which the manufacturing of a biosimilar is done may not be exactly the same. So that is where the innovation part comes into picture. How differently or how similarly can we make the product which has the same functionality and activity, purity and potency, but yet make it cost effective in terms of the manufacturing process. Because there are multiple players and it's not a monopoly, it stimulates market competition, which indeed overall brings down the price and brings in affordability to patients and access to a much bigger patient population. Once a product moves from a phase of patent into a biosimilar, that is, once it goes off patent, the cost of treatment is seen to decrease by at least 50%. And this 53% reduction in the annual treatment cost is an average that is seen across different therapies or medicines that are made in. So this is where the affordability is truly contributing to improve the patient. And in terms of sustainability in healthcare system, if the medication is extremely expensive, as such a person would be ill in order to take a treatment or be under medication. So we are adding a financial burden to an already existing medical condition. So it makes the patient and the family that is associated much more in a trouble state. So in order to ensure that it is much more sustainable, this process of bringing in biosimilars and affordable drugs is very much important. Moving on to the science behind biosimilars. So there are a couple of aspects that we come into the picture when we say biosimilars. There will be a standard manufacturing process where the cells will produce the drug of our interest, and that is utilized to characterize that product to see that its structure and function is appropriate. And then there is a clinical study which is done to confirm that the product has the same efficacy as that of the originator. Once all these studies are completed, there is a totality of data which comes in to prove that it is a biosimilar. If you want to further break down the development pathway, there are multiple steps which are involved in the journey of the drug from a bench to the bedside of the patient. So it would involve cell culture, or this is where the cells will produce the drug of our interest. So the cells are engineered, and the sequence which will ask the cells to produce the drug of our interest is inserted into the cells, and the cells will produce that and secrete it into the supernatant that would be present in the cells. Once that is produced, we have to purify the drug, and that's where the purification process comes into picture to separate out all the impurities, the cell metabolites, everything that is present in the medium other than the drug, which is our product of interest. Once that purification is done and we have a clean product, it is formulated so that the product is now stable and can be stored for a certain period, so that until it reaches the patient, and also while the patient is taking it, in that period the product doesn't degrade. So this is where the manufacturing process kind of ends in, and the next step of characterization starts in. Like I mentioned earlier, these are very complex molecules, so we need to characterize it at multiple levels in terms of structure, in terms of different functions that it can have. So we'll look a little about this in the further slides. So I'll keep it brief here. Once we finish the characterization and understand that the structure and function of the product is similar to that of the originator, then there is a clinical study also which is performed in patients, so that we can determine the efficacy of the drug is the same in the patients as well, not just in a lab setting. Once that is done, all of this data is combined and submitted to the regulatory agencies of the respective countries, so that we can gain an approval to provide this to the patient. So this is a basic journey of the drug from where it is manufactured to the patient. Moving on, as I mentioned, characterization. So here I will take an example of an antibody as a therapeutic biosimilar. And when we look at the antibody, this is the basic structure of an antibody, where you have two heavy chains, you have two light chains, and there are different regions of the antibody. And you will also see some disulfide linkages. There is a glycan structure coming into picture. So it has multiple components into it. And each region has its own function. Usually, the Fab region is the antigen binding site, and the FC region is where it stimulates, binds to the immune system of the body and will elicit some responses. So in order to understand this molecule better, what we do is two different types of analysis. One is at the structural level, the other is at a functional level. When we look at the structural analysis, the first part would be to see that the sequence or the primary structure of the molecule is similar for both the original biologic and the biosimilar. This will be the protein sequence or the amino acid sequence, which is part of the protein. Once we confirm that the amino acid sequence is same as that of the originator, then we look at the secondary structure. So because these are very large molecules, they fold into a much more complex structure. It is not a two-dimensional structure that these products would have. So they have a secondary structure which can be an alpha helix or a beta sheets. There will be loops and turns, so that they form a holistic structure. So this basic components of a structure is what is evaluated as a part of secondary structure analysis. Once the secondary structure is confirmed, then an evaluation of the tertiary structure or the 3D structure is carried out. Tertiary structure would say limit to individual chains. Say it might be just for the light chain or just the heavy chain. And the higher order structures would say consists of this whole molecule as it should appear in the three-dimensional format in a solution. So that's what is a higher order structure. So we do the structural analysis at multiple layers. Once we know that the structure is indeed intact, then we move on to the next phase of analysis. We know that it has a structure as it's supposed to be, but does it also function the same way? Does it have the same responses? Does it bind the same way to the target? So in those terms, when we look at it, we will look at the target binding capability, because these are all targeted therapeutics and they have a specific antigen to which they go and bind. So is it able to bind in the same way with the same strength to the target of interest? Once it binds, there is a mechanism of action of the drug that it will induce. Say, for example, we are looking at any anti-cancer drug. Then this drug would go and bind to some antigens which are present on the epitopes which are present on the cancer cells, and it is supposed to kill the cancer cells or stop the growth of cancer cells. So that stopping of growth of the cancer cells is one of its mechanism of action. Then there are some other antibodies which can also elicit our immune system. It can activate our immune system to identify and target these cancer cells to kill them. So we will evaluate these functions as well. Some of the biosimilars or drugs can also have undesired or unwanted effects, adverse effects. So what we also do is look at drugs and see if it is having an unwanted response. So these evaluations are also carried out as a part of complete functional analysis. Once we have this, we know that the molecule has a structure as it's supposed to be, and it functions the way it is expected to. So these are some of the data which I've taken from a publication for trastuzumab. Here on the left top corner, you will see a mirror image or a butterfly kind of a plot wherein we see the sequence of a originator trastuzumab and compare it to the biosimilar trastuzumab. And as you can see, these are mirror images of each other. The green profile and the red profile in these two graphs are mirror images of each other, indicating that the sequence is exactly the same as it's supposed to be. So this is one part of the data I am showing in as an example, which is a primary structure determination. We also look at different aspects in terms of, say, because these are complex molecules, they can be in monomeric form or they can aggregate as well. So is the distribution of these high molecular weight species same as the monomer content of the drug? So we evaluate this for the originator as well as the biosimilar product, and we are able to see here that the way it appears for both the originator and the biosimilar is exactly similar. Here we are looking at the different charge variants on the right bottom corner. So we see that the acidic species are very similar between the originator and the biosimilar. The mean is also similar, and the basic peaks are also similar between the originator and the biosimilar. So this kind of a data is what we generate to show that the structure is comparable between the originator and the biosimilar product. Moving on to the functional analysis data, as I was mentioning earlier, these are different aspects that we are looking at. This is the target binding of the drug that we are looking at. As we increase the dose of the drug, we see that it is able to bind to its target much better, and it is very similar between the originator and the biosimilar. So here we've taken the example of trastuzumab, which has a target as HER2 proteins, which are expressed on the cancer cells. So in terms of binding or identifying the cancer cells as target, the originator and the biosimilar is able to identify it equally well, and there are no differences that we see. Once it binds to the target, it will stop the proliferation of the cancer cells which are expressing HER2. So as we see here, as the increase in the drug happens, drug concentration of trastuzumab increases, there is an increase, there is a decrease in the extent of proliferation that we are able to see. If the drug concentration is much lower, the extent of proliferation would continue to be somewhere around this. But as the drug concentration increases, it decreases the extent of proliferation to a great extent. So in terms of understanding, if you want to look at it, how does it translate? Then it would be that, say the tumor sizes of say 2 centimeters, then it continues to remain at 2 centimeters because this drug is inhibiting it from growing further. In addition to stopping the proliferation of the cancer cells, what it is also able to do is kill the cancer cells by an antibody dependent cellular cytotoxicity, or what we call as ADCC activity as an acronym. So once the concentration of the drug increases, we see that there is an increased cytotoxicity that it can induce until the saturation is reached. So this also indicates that the biosimilar product is able to kill the cancer cells to the same extent as what the originator product would do. So all of this data combined says that the drug is able to identify and bind to the target cells, in this case cancer cells, and stop the proliferation of the cancer cells and also kill the cancer cells. So this gives us a total knowledge of the fact that the drug is equally good as the originator in terms of its functionality. So coming to the totality of evidence in order to establish biosimilarity, we do the analytical similarity evaluation, which involves the structure and function activity, which I was mentioning about, which will include all the critical quality attributes of the drug. So as in case of trastuzumab, ADCC is a function which is critical activity for the drug. So we will evaluate all critical quality attributes in terms of structure and function and show that they are similar. There can be some additional non-clinical testing that is carried out. Now, if for an example, if we look at trastuzumab only, it is used for treatment not just for breast cancer, which is HER2 positive, it is also utilized for gastric cancers, which is HER2 positive. So in the case of a clinical trial, we might have run it in the major population of the disease, which is breast cancer. But we don't need to carry out a separate clinical trial for gastric cancer. So that is where we can utilize non-clinical testing, wherein we use a laboratory system or an animal model where we will use gastric cancer cells or say animal models with gastric cancer and show that trastuzumab biosimilar is able to kill the cancers just the same way as innovators would. Once we establish that the drug is able to induce killing in the same way for all the indications, we also have a clinical data coming in from the trials in the main study, which will give us the PK and PD data. So PK is nothing but pharmacokinetics and PD is nothing but pharmacodynamics. So when a drug is given to a patient, there is a two-way reaction which happens. In one, the drug goes and acts on the target or the disease, and the body or the host also identifies that there is some different product which has come into my system which is foreign, and it will also have some reaction on the drug. So this evaluation is what we do with clinical setting, where we get a clear understanding that the way the body behaves when a drug is provided and how the drug indeed works on the target cells is similar in the case of biosimilar as well as the originator product. We will also do some, because these are large molecules, they are capable of inducing immune system to identify it as a foreign molecule. So there is a possibility that the host system or the person who's being given the drug can develop an anti-drug antibody which will go and bind to the drug and clear the drug out of the system, because it knows that this is some foreign particle or foreign molecule which has come into the system. And some of these may also go indeed and bind to the antigen binding site, which will in turn become neutralize the drug and will block the activity of the drug. So we also check for ADA and NAB as a part of the overall evaluation. And we see that in case of biosimilars, it is also inducing a similar level of ADA and NAB as the originator would be making it. Once we have the clinical data which says that it is efficacious and safe, analytical similarity information which says that the structure and function is appropriate, and the non-clinical testing data which says that it can be extrapolated to all the other conditions in which the drug is recommended for. This integrated conclusion is what establishes biosimilarity, and this integrated conclusion is what is called as totality of evidence to demonstrate that the product which has been made is truly indeed biosimilar to the originator. So we were talking about clinical studies till now that, we treat the patients with the biosimilars. But there is a recent trend that is coming up that there are phase 3 waivers which are given by the regulatory agencies. This is coming from a huge amount of data over the past 2 decades where a lot of biosimilars have been approved. And it has demonstrated that although a phase 3 trial has been performed, the information that gives us a confidence to go ahead and call something as a biosimilar can be done with much lesser information. So UK regulatory agency, which is MHRA, has said that they do not require any phase 3 studies. EU medical regulatory agency, which is EMA, also says that many of the phase 3 are not necessary. The US FDA has now a draft guidance which is telling that switching studies and phase 3 is not required. And Indian regulations are also aligning with the global trend in terms of phase 3 waivers. So overall, what is the impact of this phase 3 waiver is that the development duration can be much faster and the cost of development can go down significantly, because one of the biggest components in terms of cost for a biosimilar development is the phase 3 clinical trials, which are usually quite huge and comes close to 100 million just for the clinical studies. Based on the recent regulatory information, the design and development of methods used to characterize and the analytics, the data analytics that are associated with it, is what should be utilized better to demonstrate a biosimilarity and comparability of the product of the originator and the biosimilar. So this indeed decreases the burden on the manufacturers as well to bring down the costs much more easily, and allowing a much more better market competition and patient affordability coming in, because if the cost of development goes down, it's much more easier to translate the same thing to the patient and bring down the costs. So overall, if you look at what is the biosimilar journey in India, till now we understood that how we can get an manufacture a biosimilar, how we can get an approval, and how does it help patients in terms of affordability. What's the story of that in India per se? So India has got a very good manufacturing capability and a highly skilled workforce. So we are able to produce these drugs at a much more affordable price. And the government policies are much supportive, and the regulation boosts the growth and innovation in terms of pharma sector overall. In terms of the global recognition, there are multiple Indian firms who have gained international approval in US, be it the US FDA approval or the EMA approval for complex biosimilars. This demonstrates our capability and advanced skills that we are indeed capable of making any complex drugs which are used for treatment of patients. So the overall outlook in terms of access and affordability in India would be to make India a major supplier. Today, India is indeed a big supplier of quality and cost-effective biologics worldwide. We are one of the biggest suppliers of biosimilars for US as well as EMA. So in summary, what we have understood today is that biosimilars are affordable and accessible innovation. And it's a balance of affordability and innovation, which makes it much more patient-friendly. It's built on a scientific stepwise approach so that there is no compromise in terms of the scientific rationale which is associated with the development of biosimilars. In terms of the regulatory frameworks, it is converging globally what the FDA says, what EMA says, and what Indian Swissco and all the other regulatories. They're all converging and understanding that this is the need of the hour and the frameworks that is required for any manufacturer to make drugs would be similar. India has indeed now become a global factory for the biosimilar production. Just as a takeaway, biosimilars aren't just alternatives, they are indeed the future of sustainable healthcare with targeted therapeutics, with them being the targeted therapeutics. Thank you. Hi, Shuhini. I think you're speaking.
Shuhini, ma'am, you're on mute. Yeah.
Never mind. Sorry. So thank you, Malini. Thanks so much for that insightful presentation. It's clear that biosimilars are not just cost-effective alternatives, but they play such a vital role. They have a vital pathway to sustainable healthcare in India and in the world for sure, and which is built on rigorous science and innovative development. So they truly represent the future of affordable global healthcare in that sense. I have many questions, and I'm sure the audience does too. So I'd like to begin with my questions before we make it open to the audience. Malini, as you know, as you mentioned also, unlike small molecule drugs, biologics are large and structurally very complex. And this complexity kind of shapes the development of biosimilars that you walked us through, right? So which brings me to my first question. Why is it not possible to make exact copies of these large molecules?
As I was mentioning, these large molecules are made in living cells. And the sequence that we do utilize to make these cells are very similar or exactly the same. But we know that we work with a living system. If you look at ourselves also, the way our body behaves, say early in the morning versus later in the day or towards the evening, we are behaving differently. Why? Because there are certain factors that are associated in terms of, say, the nutrition, the rest. So all these factors are affecting us. Therefore, we are able to see the same translating into our behavior being different. Now, if we take this as an analogy to the cells are also having a environment in which they are growing. We are adding all the nutrients that is required for the cells. So when we add all the nutrients to the cells, they are happy, they will grow, and they will express the protein of our interest. Now, when we say express, so we have given it the code which is required for expression. and it will produce the amino acid sequence in the same way. But because many of these large molecules are prepared or manufactured in mammalian cell lines, there is a much more complex system which is associated with it. Once the protein is expressed, it undergoes a post-translational modification within the cells which are producing it, and that is where many of the differences do come in. So how are the metabolites that it is produced, how much of glucose it is utilized, how much of CO2 it has produced, all these, because this is not a whole organism kind of a thing where we know it's very well fixed. I mean, we are not yet able to mimic a whole organism biology, but here we still try to control the environment. So there is still some phase wherein, as the nutrients decrease, there will be increase in the amount of metabolites, the CO2 production will also go up. So these are the smaller factors which will indeed make it different. And the other part of this is that these host cells that we are talking about, they're not exactly the same which the originator would have used it. So this also adds in a small amount of difference, like the way, both of us are there, both of us are women, but we're still having some minor differences between each of us. So it's exactly that way. So in terms of output, it may appear the same, but there are some minor differences in terms of the way we would choose certain things. And that's the exact same way, if I have to bring in an analogy, that cells are still different. They're not the same cells. So we do see some differences in it.
Absolutely. I think the human body itself is such a complex
Yes.
and organization of cells that I'm sure that kind of mimics what we are trying to do here. Just a quick question again. So what is the developmental timeline for the average for a biosimilar?
So as of today, I think the initial biosimilars which was made in took a much longer period because everyone was learning what is required for a biosimilar. So initially it used to be around 7 to 8 years. But as of today, I think 5 years is what is a realistic target to make any biosimilar. And this is going all the way from starting in the conceptualizing that we will work on this biosimilar to getting a regulatory approval. That whole process is around 5 years.
Wow, but that's still remarkable sort of improvement in the timeline.
Yes,
right?
yes, indeed. If you look at the originator biologic that they would make it,
Right.
it will take around 15 to 20 years to bring that product into the market, whereas a biosimilar will require around 5 years to bring it into the market. So it makes a huge difference in terms of time.
Absolutely. So another question I had was, we talked about since biosimilars cannot be identical copies of the originator biologics, right? Maintaining consistency and demonstrating high similarity, therefore, requires rigorous testing and controls, as you mentioned, throughout the development and manufacturing. Now, when it comes to ensuring the quality and safety of biosimilars, what techniques are typically employed for batch release? And also, as a follow-up question, I wanted to understand if you could, I wanted to ask if you could elaborate on why validating both the manufacturing processes under GMP and the analytical methods used for batch release testing are so critical in the biosimilar development pathway.
Right. So if I break that question down, the first part would be how do we assess that the batch is fit? So that's where I think, we do this even for normal reagents that we procure in the pharma, that we have a certificate of analysis which says this is the purity, this is the total content of the whatever molecule that we are buying it. Now, because these are much more larger, we have to perform a much more wider list of tests. So some would be the structural analysis aspects that I was showing in earlier in terms of, say, the charge variance, the size variance, the glycan content as well. So there are multiple structural analysis which is done in, and there is also a functional testing which is done in, like I was saying, say, in the case of trastuzumab example that I was taking. There's a HER2 binding, there's a proliferation inhibition assay as well. So there are multiple things that are tested in, and there's also microbial testing because we are giving to patients. Most of these drugs are either IV or subcutaneous injection, so it's directly going into the system. So they should not be, they should not cause any infection by themselves. So there is a microbial testing to ensure that there are no foreign particles, as in microbes, which are present can trigger an infection. Now, when we are using these tests also are having their own inherent variability. So that's where the validation comes into picture. So when we say a product has a 50% activity or 80% activity, how do we know that the method is capable also of picking something 80% or 50%? So we stimulate the system in such a way that we can ensure these methods are capable enough of detecting any differences. Our target would be to have 100% activity and in terms of structure to be bang on to the expected levels. But because these are all systems which have come in from mammalian cells, we may see some differences in terms of the product. So that's where the methods which are utilized to evaluate should be something which we have confidence in, that they are indeed capable of picking up any differences that do occur in. So that's why we have to have the methods validated. There is also a requirement for the process to be validated. Now, if you're giving a drug to a patient today, now tomorrow when we manufacture the same thing in a different batch, how sure are we that the output or the product that is derived is the same and the quality of the product also remains to be the same? That's where I think the process validation also comes into picture. We validate that the product that we are making using this particular manufacturing process gives us the same output each and every time. So that's where validation is required for both process as well as the techniques that are used to release as well.
Thanks so much, Malini. Thanks for so patiently explaining that in detail. I think I'd like to now open the floor to the audience for a quick round of question and answers.
We have two questions in the chat box, Malini, for you. The first one is, what does interchangeability imply? And the second was, who are the primary stakeholders involved?
Right. So interchangeability is something which is very specific to the US market. When we talk about interchangeability, what we exactly mean is that I have an originator product with me, one which is present here, and I have a biosimilar. And I can interchange and give it to the patient. So the patient was originally on the, originator product. And now today there is a biosimilar available. Can they switch from originator product to a biosimilar product is what is about interchangeability. So the guidance around interchangeability is different with the different regulators. US used to have a very specific interchangeability studies to be carried out to get that claim of interchangeable. Whereas in EMA, it's a default understanding that, since you have done so many studies, you don't need to show that I can switch between originator and biosimilar. So in terms of those studies is where the difference lies in. It's more US-centric aspect that comes into the picture when we talk about interchangeability. Moving on to the next question, I think it was on who are the stakeholders in this study in the biosimilar perspective. So there are multiple people who come into the picture. One is, of course, we as manufacturers of the drug. That part I think we've already covered in. The next part who play a important role is the physicians who will prescribe the drug to the patients. So they need to understand that what is the efficacy and the safety of the product, because they are the face of the drug to the patient. So they need to be sure that the product that they are prescribing is safe and efficacious, and they can indeed prescribe to the patient. So that's one party in the one of the stakeholders in the biosimilar perspective. Then we have pharmacists. So although say the doctor prescribes, and if the pharmacy does not have the product, the biosimilar product, then the accessibility becomes a question. So that's where I think again it's important to have pharmacists also aligned that there is a substitution product which is available. And in different countries, the pharmacists play a different role. So in India, you just go with a prescription and you say, I want this drug. If the doctor has prescribed you a biosimilar, then the pharmacist would directly give you a biosimilar, which the doctor would have prescribed. But if you go to US setting, the pharmacist is also a big stakeholder, because the doctor would prescribe, say, for example, trastuzumab is what the doctor would prescribe, but the doctor would not say whether it is Herceptin or any of the biosimilar molecules that are available in the market. So the pharmacist determines whether the product which is given to the patient is the biosimilar or the originator. So their understanding and educating and informing pharmacists about the availability of biosimilars as a substitution product is very much important. And they are also the face, again, who interacts with the patient. So they will also be counseling the patients that this is equally efficacious in terms of the way it treats the condition. The third stakeholder, if I have to say, is the main stakeholder, who is the patient, because they need to be educated and made aware of that these products are indeed safe and affordable and much more accessible to everyone. It's in different regions you have different settings. So the economy and the economic state of the person, how financially sound they are, how much of an insurance support do they have, and so it plays a very big role there in terms of the patient, that the patients should also have a confidence that indeed biosimilars are as good as the originators, and there is no difference and nothing for them to worry about or be concerned about. So that's where patient is one of the important stakeholder. And the last but not the least stakeholder are the payers. These are formularies or insurance folks who actually pay for the product. So at least in the US, it's that these are the people who kind of decide that, as an insurer or a payer, I will endorse biosimilars because it costs me also lesser and I can provide it to much more patients. So these are the people who actually work around with the money part of the drug or the costs associated with the drug. So that's where I think they work more on cost containment and inclusion, because if they don't include a particular biosimilar into their list, the patient, although may be aware of biosimilar product being available at a much lower price, they might not be able to access it because their payer will not provide that one as a option for them. So these are key stakeholders in the biosimilar business, where we need to have them aligned, that of the availability and the affordability of the biosimilar.
Thanks, Malini. While we wait for some more audience questions, I have a quick question. I know, and this is about clinical trials, which is kind of close to my area of expertise. You talked about phase 3 waivers and, the recent regulatory trends around them, EMEA and India, of course. So, the question is that while you may get a phase 3 waiver, you would still need the other phase clinical trial studies to continue, right? I mean, those are still essential.
Yes, it's still essential. So it's not that we do not require any clinical studies at all, but the phase 1 is something which still remains as a requirement, but it's a much smaller population, and it may be also carried out in healthy volunteers. So The cost associated with phase 1 is much lesser, and it gives us essential information in terms of the safety of the drug. Because these are biosimilars, maybe a few years down the lane, we will get confidence that biosimilars are also safe, like the way we've now moved from phase 3 essential to a phase 3 waiver. There may come a time in the future where phase 1 is also waived off. But as of today, they remain essential part of the submission and requirement for approval.
Are there, do we have some more audience questions?
No, we don't have as of yet, ma'am. Yeah.
Okay,
Yeah, we have Rohan. We have Rohan who is raised. Yes, Rohan, please ask.
Hello. So my question is that to what extent should similarities be expected when differences arise during characterization, and how should these differences be managed?
So differences can exist at certain levels. Uh, that's where we do a characterization. So when we do a structural characterization, we may see that there are a few differences that do exist in terms of the structure. But does that really translate to a function is what we try to understand. So that's where the functional testing helps kind of de-risk the differences that may be seen with structure. Like I said, these are inherent variabilities because we are producing it in mammalian cells or living organisms. So there is a possibility that there might be slight differences, but are these differences truly meaningful is what we try to do. So structural differences can be evaluated in terms of functionality. Does it affect the functionality or not? And that can de-risk the structural aspects. But say if we see some differences which are not just structural but also have functional differences, then there are other testings that can be carried out in terms of, say, immunogenicity testing. That does it really cause a very different immune response? So all of these testing can, as of today, be done in vitro and does not necessarily mean we have to go to a patient population and do a clinical study. So there is a possibility that we can utilize immunogenicity testing and show that although there might be some differences, these do not really have any effect on the product. But if you look at it, this is from a industry personal perspective, but from a regulatory expectation would be that, the ideal target is not to have any differences in terms of structure or function. The functional differences raise a much bigger flag because it tells that it is impacting some function or activity for which the drug is actually given in. So there is certain amount of risk involved, and in fact, if this is done at early stage of the program, you can go back to the bench and see what you can do changes to the process so that the functional differences are removed and it functions the same way. So that's how you can manage the differences in terms of the product that may arise during development phase.
Thanks, Malini. We are almost at the end of our time for this webinar, but before we close, I'd like to ask one last question. With advances in digital technologies, transforming healthcare, and this is something that's so pertinent to what's happening around the world, how are AI and machine learning being utilized in the biosimilar development process?
I think that's a question all of us are also trying to understand and utilize. AI and ML are the buzzwords which are going around today. So one aspect which I do see which can be utilized very immediately is in terms of, say, assisting the documentation preparations. Like, say, as scientists, we conduct experiments and we know that there is a certain result that has come out, but we have to document it in a certain appropriate format. So in terms of executional inputs, like, say, if I'm adding X amount of a drug, if there is a way in which we can bridge the technology that a pipette is connected to a server or a system, that it will detect that I have added indeed 10 microliters. So it will do all the rest of the documentation accordingly, that if a 10 microliter of the drug is added in, this is the output that we are receiving. So that is where AI can help and into the picture. The other part is, once we have all these results, we have to put everything together to say that the structure is fine, the function is fine, there are no other risks. So building these documents, when we say we submit something to the agency, these are huge dossiers that we prepare in. I think that's where AI and ML will come into a picture very soon in a big way. They will definitely help in. Another place that I can think of is pharmacovigilance. Now, pharmacovigilance is more once a product is released into the market, if there are any complaints or any risks that have been seen newly. How do we put all of that together? If it's a manual system, there is a possibility that we miss something, or something gets overseen, something is not responded to. So that's where I think today pharmacovigilance is also a place where AI and ML can bring into the picture, that if there are any queries raised in terms of pharmacovigilance, AI may be able to answer that directly with the available data and information at its end. You're on mute, Shohini.
Shohini,
Shohini,
you're
you're
on
on
mute.
mute.
So sorry, I've been doing this today, but I said that's very interesting, and I think there might be even other uses of AI ML which you've not even encountered yet. I think as we go ahead, it might be even more, practical uses of AI ML. But I think that brings us to the end of our session today, looking at the time. So on behalf of Ahmedabad University and everyone here, I would like to sincerely thank Malini for, again, generously sharing her time and sharing such valuable insights into the science, the development pathways, and of course, the future of biosimilars. Thanks, Malini. Her perspectives have not only deepened our understanding, but also highlighted the broader impact of biosimilars on healthcare access and innovation. So I'd like to thank our audience also for their engaging participation and for being with us. If you have any more questions about our session today or about the university, please do contact our admissions team and schedule a one-on-one call. With that, I formally close today's webinar. Thank you once again, everyone, and I wish all of you a wonderful day. Bye, Malini.
Thank you, everyone.
Thank you. Thank you.